Click for IB Biology. We're looking at the so-called lipid bilayer and the molecules forming the cell membrane. There's a blank PDF of this presentation down in the description. There you'll also find 10 questions with answers, and there should also be an AI prompt structure that you can use to create questioning and self-assessment for yourself. So the structure of the cell membrane is critical in understanding almost every aspect of biology and the way in which living things interact. Either they're cells interacting with each other or the cell interacting with the environment in which it is found. And what we also find is that the so-called cell membrane, illustrated here by this double layer that we can see here, is often a barrier between two types of solution: the extracellular fluid, the external environment, and the internal cytoplasmic fluid, both of which heavily feature the presence of H2O, water. And the principal molecule in forming cell membranes is the structure which I've got here. And we're going to call this a phospholipid. And there are essentially three components to this phospholipid, split into two. At the top end, we have a so-called hydrophilic head structure. The word head is usually used. This is a polar, a charged structure sitting at the top here. And the word hydrophilic, let's think of that as water-liking. Attached to the phosphate, there are two hydrophobic tails. These tails are both fatty acid tails. They are non-polar, they have no charge, and they are described as being hydrophobic, which is to say that they repel water. Students would do well to return to this video and indeed all of cell membrane structure after you have studied the biochemistry of proteins, lipids, and carbohydrates. So this molecule, the phospholipid, which makes up our cell membranes, is described as being amphipathic with both negative and positively charged regions, and indeed, of course, the tails with no charge at all. One of the concepts that we'll be dealing with is the idea of the spontaneous organization of molecules, and this relates to the concept of Gibbs free energy and the idea of an exergonic process. So essentially what we're going to be seeing is where molecules will organize themselves into the lowest possible energy state, and this is described as spontaneous organization. So if there's a way to organize yourself so that you are at the lowest energy orientation of the molecules, that's what they do, spontaneous organization. So let's look at our lipid bilayer. So as I previously mentioned, at one end of the phospholipid we have the phosphate group, and then at the other end we have this fatty acid chain. I've just drawn one here for clarity, but in fact there would be two, so two fatty acid tails there. And then we remember we characterize the head area, the phosphate area, as being hydrophilic, and the tails are hydrophobic. Delta G refers to Gibbs free energy and our prediction that these molecules are going to organize themselves into the lowest energy organization possible. And that begins by them turning the tails towards each other, like we have here. So the hydrophobic tails are going to be at the center of this structure with the phosphate heads around the outside. And this presupposes our description that we're in a water type environment. So the phosphate heads around the outside, they want to be in water, but the tails don't, and so they align themselves. Now this structure can equally be reformed into a more elongate structure like this. So you can see the phosphate heads along the outside here. That's fine. Remember, out here is water. Remember, here is water. And so the molecules are organized. So the phosphates are quite happy to be in water, energetically speaking. But the tails want to be with themselves. They don't want to be in water at all. They repel the water. And so we get this layer D, where it's a flattened layer structure. Now all we need to do is to extend this all the way around, and for it to grow a little bit, to get a little bit larger, and we end up with structure G. And we're moving now more towards a typical cell structure. And this structure that you're looking at is indeed at the core of all cell structure, which is the what is famously called the phospholipid bilayer. And the only thing that we need to emphasize here is that the phosphate heads are on the inside, the phosphate heads are on the outside, and that's with water, the extracellular fluid, that's with water, the water of the cytoplasm, and the lipid, the fatty acid tails, they're together. That's energetically how they prefer to be. And in fact, what this does is they will form the actual barrier of the movement of substances into and out of cells. Just one extra thing to add to this is that try to remember that this is not a circular structure, this is a sphere. And so, although we're in cross-section to show you the structure, we need to be thinking all the time of a near spherical structure, phosphates on the outside, phosphates on the inside, but this double layer of fatty acid tails in between. And historically, this was the first conclusion that scientists came across. In fact, they were called Gorter and Grendel, 1925, and this was one of the earliest models presented on cell main structure. But that would be replaced later in 1972, well, progressively replaced, and finally replaced in 1972 with an altogether more elaborate model as the experimental evidence accumulated, so that we could update our hypothesis about the structure of the cell membrane. Links to the next video, which I think is on the concept of simple diffusion, arising there. Subscribe to the channel, please. And there on the right hand side is the playlist for this topic of the syllabus.